Welcome back to the biology show, and today we're hitting DNA biotechnology part 2. All right, let's start with gel electrophoresis. Very effective type of biotechnology. So what this type of technology does is it's used to create like a DNA fingerprint, and how our fingerprints are unique to only us. Our DNA is unique to only us. So no one has the exact same set of fingerprints, even identical twins. So we can use them to identify people like criminals or things like that. They use them in paternity tests, and our DNA is unique to us. So how this process works is you have different DNA samples. So different DNA samples are exposed to what's called the same restriction enzyme. So they're exposed to the same restriction enzyme. They're put into this is called a DNA gel electrophoresis, and we're going to do this in class. So there's these, quote, we call them wells, and then we put the DNA in here, and there's charges here, a positive and a negative charge, and these different fragments of DNA are going to run through this gel. So they're going to run from the negative down to the positive. And so a portion of that, not like a whole vial, but a portion of that is pipetted in here, and then and this is like gel. I mean, it feels kind of like Jello. And and then you can see how the strands of DNA are going to they're going to move through this gel, and eventually going to end up with something that looks like this. The shorter molecules are going to run farther, and the longer ones are, I mean, it just makes sense. If it's easier to move, it's going to move farther in the same amount of time. All right, so the different DNA samples are exposed to the same restriction enzyme and the cutting enzyme. And what this does is it creates what's called a restriction fragment length polymorphism, RFLP. All right, so restriction fragment length polymorphism. What these are fragments of DNA that have different lengths that were created using these restriction enzymes. So you can see that in here, restriction fragments. So we've used restriction enzymes and we've cut them into fragments into different lengths, and polymorphism, many different shapes. Okay, and so that's what we're doing here. We're cutting the DNA into different fragments. So the DNA RFLPs are loaded into an agarose gel. All right, and here's the gel right here. Here's what it looks like when it's completed. So it's loaded into the gel. The electricity is turned on, and again, DNA is negatively, I don't know that you know this, I can't say again, but DNA is negatively charged because of that phosphate backbone. Remember the PO, and then it had the negative, the phosphorus and the oxygens. So it's going to be negatively charged, and it's going to be repelled on the negative end, on the black end, and then pulled by the positive end, the red end. So like here, the positive end, because it's negatively charged, it's going to be repelled here, but it's going to be pulled down through here. And those RFLPs are going to separate according to the length or the size of the fragments. So here's your negative end. It's going to move down here to the positive end. It's going to separate based on the length of those fragments. Obviously, big pieces move more slowly. If you think about it, in terms of football players, the big, heavy linemen don't move as fast as the small, light running backs. Doesn't mean that they can't run. It's just harder to move all of that along. So, all right. And the other thing that you need to know here is the DNA fragments are also stained for ease of viewing. So we can see these in here, so we stain them. And then ultimately what happens is we create this band. The DNA bands create this fingerprint, and this is only you. All right, now the Human Genome Project. I've referenced this several times in class. Just a fascinating, amazing thing that's been done. The Human Genome Project, sometimes called the HGP, this project was begun in 1990 and ended in 2003. And um, and what happened is the project mapped out the entire human genome, the entire DNA genome nucleotide sequence for all humans as a species. The human genome contains approximately 40,000 different genes. So now, excuse me. Now you also have to remember that rearranging the exons and how that allows humans to create way more than 40,000 proteins and enzymes. So we think about 40,000 different genes, but remember when we ligased those back to, I mean, when we spliced them back together after the spliceosomes took out the introns, the exons were spliced back together, and they can go in any shape. I mean, they can go in any position. So when they go back into a different sequence, it's going to make something completely different. So it's going to make way more than 40,000 proteins or enzymes, okay? Now those 40,000 genes only make up about a third of the total genome. So only 3% of the entire genome. So in other words, 97% are control sequences or introns or quote spacers. All right. Amazing project. It was this tremendously collaborative effort. It was not one scientist from one country. It was from all countries. I mean, they all worked together on it. All right. So, to talk about gene therapy in order to treat diseases. So, gene therapy, what this does is it uses a virus in order to introduce a new gene into the body's cells' DNA. So in order to introduce new material, so here's this DNA, it's put into the virus. The virus inserts the genetic material into the human cell. Now, when we talk about somatic cells versus germ cells, somatic cells only affect you. These are your body cells. Germ cells affect future generations. That's your sex cells. And so somatic cells are only going to affect us. And but it's kind of like working out. If I work out, it only affects my body. If I become a bodybuilder and get super buff, it's only going to affect me. It's not going to affect my children or my grandchildren or anything like that. All right. New DNA-based medicines. We have a whole area in pharmaceuticals where we use this DNA technology. This helps with creating new medicines, vaccines against diseases. I mean, obviously we have Ebola that's been very prominent in the news, and that's exactly what they do. And we're hoping that even cancer in the very near future, the technology is used. Criminal investigations, we use DNA fingerprints of suspects or paternity or maternity testing. Environmental cleanup in the Gulf or anywhere, environmental cleanup. So bacteria is used to process the human sewage in wastewater treatment plants. They actually use bacteria in there. They can also make bacteria that can help clean up oil spills or break down plastic by eating the oil compounds. Those were used in the BP Gulf oil spill. They used those different bacteria that were genetically modified. We have organisms that help clean up heavy metals such as mercury from mining areas or waste collection. In the area of agriculture, we have a lot of engineering done to produce more and larger foods. Genetic engineering organisms are able to produce hardier food that are easier to transport. So they're going to last longer. They're going to ripen slower, maybe. We have organisms that produce healthier foods or organisms that can produce food during winter. So they're winterized. You can see here's a different color. We can change the color. We can all kinds of things. All right, with livestock, we produce, look at the difference here in these fish. And so obviously we can produce organisms that are meatier or maybe leaner. So it's going to have less fat, so that's healthier for us. Maybe organisms that are disease resistant. And so we can produce all kinds of different organisms. One more thing I want to talk about is this concept of transgenic organisms. This is where we combine DNA from two different organisms they're combined to make one organism that has traits from both. And let me give you an example of this. Right here, you have broccoli and we have cauliflower, and together we make broccoli flower. And so it has the traits from both parents, and those traits are going to be passed down through reproduction because the traits are in the DNA nucleotide sequence. Um, and so you end up with these different characteristics, and it can have a very positive result. Okay, that's it. Great job. Dun, dun, dun. We are done with molecular genetics.